UCSB, we RET2 programs. It's my second year. And this is just the beginning slide, but my title, I decided because this is a poll in my curriculum, is about global climate shifts. Because it's in all of the NGSS standards, but it doesn't ever seem to get taught because it's way at the end of our units at the end of the year, and people just go, oh, I'm not going to do it. And plus, maybe we just don't know how to do it. So I came up with this, where there is a very, very large struggle with the students and with teachers too on plain evidence reasoning. And so as I go through this, you will start noticing that I keep having the same format for my plain evidence reasoning, but I switched it around thanks to my colleagues who said, maybe evidence reasoning in plain would be a lot better for a student's mind on how to get through this. So I like climate shifts over time. My background is in geology. So I have this picture right here, and it really does show what the climate has done all through geologic time. And even if you can't see all of the little pieces, you can see that definitely there were some shifts. And the shifts have actually happened naturally, but maybe we are kind of speeding it all up, right? And that's what the point of this whole thing is for my students. A little bit about me. Again, my name is Laura Branch. I've been teaching at Rigetti High School for the past 25 years. Love Rigetti High School, but now I'm always going to call it Regretto now, because right now I call it Rigetto, but Regretto is really awesome. I was recently named the 2025 Santa Barbara County Teacher of the Year, big giant award that had a lot of applications and interviews and whatnot. So it's a pretty cool thing to have on my resume myself. I currently teach a concurrent enrollment physical geology course. I teach AP environmental science, and I teach a chemistry and earth systems course to juniors. All kids have to take that class at my school, and it is very NGSS oriented. So there's that. And this is me in the rainforest taking data. And it's not anything to do with geology in the rainforest. It was all biology, so it's kind of interesting. Last year for my RET1 experience, I was looking at the absolute age dating of carbon that was buried in ash layers in the Monterey Formation. And with all of these things called zircons, which I'll get to in a minute, we were looking at what is the ratio of uranium and lead within these zircons. And based on that, you can tell the absolute age date that we're looking at. So when I would come into the lab, this first off, all geologists tell you where the site is where this was picked up. So these are ash layers within the Monterey Formation. My mentor, Alil, he collected them all. I brought them into the lab. I had to grind them up. I had to go and separate all of the mud, speed up that erosion process, get all of the really, really dark stuff, the really dense stuff. And then from there, I had to pick out these little tiny sand-sized pieces. You can't even see it right there, but these are what those zircons look like. And putting it all together in a stratigraphic column, which is a geology term, we were able to figure out the absolute age using these zircons. And the conclusion that we came up with, and this is what little zircons look like, we came up with this idea that the absolute age for the Monterey Formation was 15 to 17 million years. MA means millions of years ago. And so we were able to look at that. And the secondary part of my mentor's PhD, which he finally got just this last year, was that there was something called the Miocene climatic optimum. It doesn't roll off my tongue very well, but essentially what that means was that there was a warming trend in the Miocene 15 to 17 million years ago. So the connection that I came up with between the 2 years is I had some skills that I took away from last year, which was how to pick zircons, because I didn't have any clue. And then I found out that you really need to be able to pick these little tiny things with very, very small little tweezers. And I knew about the uranium iron or the uranium lead age dating. I just didn't know how the process would work. So I was able to gather the data that was needed to do this absolute age dating. And then when I started to really think around May, June, and now July, what are my driving questions for this RET2 project? And what I came up with is I wanted to have the students look at what is the specific evidence that is used to support the idea of a global climate change. Because it's a hole in my curriculum. It's like I didn't really know before this project what I was going to do with this. And then also my main thing is, since it's a weakness and if the students really struggle with it, is how do students use evidence and reasoning to make a scientific claim? And with that, we can look at what the warming and the cooling trends are going to be doing. So currently, because I have to teach this before we get into our April presentations, I'm going to be putting this into my concurrent geology course. So this is kind of the sequence that I came up with, where we have a phenomenon, and then we're going to go heavily into geologic timescale. Since it is a concurrent class, we are college level, so I can use big giant words, and really look at where are we in the grand scheme of time, right? So the students will figure that out. And then you will go through all of these other labs that you're going to be seeing in just a minute. When I get to April, May-ish at the end of the year, I'm going to be able to go and put this again into my NGSS chemistry class, where then we just look at the phenomenon, and then we go and we look at different types of data that that a scientist would actually take out for climate change. And that one would go about 9 days for 50-minute periods, which I think is doable. The NGSS standards, we hit a lot of the earth ones. I'm an earth science person, so I kind of bring to the table at my school and my chemistry PLC, I bring all of the earth in. I'm sure that there are some physical science ones in there, but I chose the earth because nobody ever chooses the earth except for me. So we're going to be looking at the Earth's early history. We're going to look at how is human activity affecting Earth systems. We're going to be looking at how is the energy from the sun coming into the Earth's atmosphere, how is it leaving, and what type of effects are going to be happening, especially with climate change. And we're going to be looking at the forecast of what could happen with climate interaction on the Earth. Like how is this all going to interact? It would just have Hurricane Beryl, which was literally barreling down as a hurricane category 5, right? And so the students will be able to start seeing that, yes, these are being affected by climate change. This is one of my new slides that I think is pretty cool. The NGSS claim evidence reasoning, it is in our standards. And what it says is that first, you're going to defend your claim, and then you're going to have a reason why the evidence works for that claim. And what I found is, and I think a lot of other teachers across California and whoever has adopted the NGSS, is that the struggle is real with the students because they don't know what to do with this. They don't even know how to make it claim, right? And then you're setting them loose, and you're like, I don't get it, right? And so they just sit there and they're on the phone or whatever, like trying to look underneath their desk with their phone. And so my big concern is how can a student write and defend a claim when they haven't even taken that evidence, when they haven't even looked at the data? How can they do that? So what I came up with, and you're going to notice during this whole presentation, is that I always have the same format for what Bill came up with, which is ERC, where my husband has eggs, rice, and cheese. I keep thinking about this, right? I'm going to say eggs, rice, and cheese, but it's not. It's evidence, reasoning, and then infinite claim. And so what I came up with is that I'm going to be giving the students either a question or a prediction or a hypothesis or a statement. Then they're going to go through and they're going to collect the evidence. So we have evidence with hints, because I have a high language learner population, hints on what type of evidence that we might be looking for, and then they can write it over here. They can give a reasoning on why they think it's there, and then they can come up with some sort of a scientific model. So as we go through this presentation, you're going to be able to see this type of thing. It just might have a different statement, or it's a hypothesis or some sort of a claim. The phenomenon, which we will be getting into in just a minute, of course, it doesn't help right now, but we're going to get into after I'm all done with my presentation, is going to be what is the evidence for global climate change. And I learned during this RET2 program, after a lot of frustrations, how to make a very, very nice, clear ice core. So, so we have teacher-made ice cores that are relatively clear, and some of our data is already coming out, so it's kind of cool, right? But that happens in real science. And so we have all of this data that can be collected, and then it was determined during this to use real seeds. So we are going to put them outside my classroom so that they can go and see what exactly is something called a glacial erratic. How does a glacier melt, and then all of these seeds come up and start growing, and you get something called succession. So that's an extension part of this whole presentation, or the extension part of the video. So when we come back to this, you are going to be doing this, where you're going to be looking at pom-poms, looking at size, looking at colors, looking at different types of seeds, and you're going to be trying to find what is the evidence, what is the reasoning for your evidence, and they're all different here because I made them all different, and then try to put together some sort of claim with talking with each other, which is kind of like a whole scientific community, which is really important too. It's an important skill to have. This is what an ice core actually looks like when it first comes out of the ground. So the second lab, or just lab number one, because that was a phenomenon, is the geologic timescale. I would do this in my geology class, where we are looking at not only how long is the age of the Earth, but also what happened. Why is there suddenly a hot climate in the Mesozoic? Why all of a sudden is there a cooling trend? And it ends up being mass extinction. And so students will be going through this. They will be mapping it out. They will have learning about looking at numbers, putting in scientific notation, looking at scaled metric equivalents, and then going through and doing this. And my thing is now having the students go through and look at this model and say, ooh, what's going on right here, right? What type of data can we look at? The second lab that I have for them, and you'll notice that here is the format for our ERC, and I give them the statement, is there a global cooling event, then, or if there was a global cooling event, then the sea level would get lower due to more ice freezing at the pole. And they have to go and figure out this data, what evidence, what kind of data would show that, and is it true, right? So they're going to go through all of this. And these are just two of the pictures from this lab. I think that there's like 5 different things that these college level students would do, where they're actually going to go through. We've learned about the different types of layers of rocks. We've learned about relative age dating. We've learned about the rock cycle, what all of these are, and now they can figure out based on this, how is this deposited and what are they going to do with this information. This one right here is called a lithofacies map. So they take some data, they have to go and draw things, they have to color, and then based on the map now, instead of a cross section, they have to figure out, would this be a sea level, it's called a transgressive sea, where it increases in elevation, or is it a regressive sea, that there's some sort of a global cooling event where the sea level is actually getting lower, because all of the ice is going to be at the course of the Earth. Not the course, at the pole. This lab number 3 is just a data set. We're going to be learning about, we're going to be learning a lot about Pleistocene, because that was the time where there's something called Pharengia. It was a cooling period. I just went to Yellowstone, learned a lot about it, on when the large mammals and humans came from Siberia area over this one little path called Pharengia, and then they came over to the North Americas and the Americas. So for this one in Pleistocene, this is one of the coldest times on Earth, where we have time, we have carbon dioxide data, and we have temperature variance. So this one works on skills where students are actually graphing, which is a great skill for them to have. And then based on their graph, then they're going to go through and they're going to figure out all of their evidence based on there is a correlation between carbon dioxide concentration and temperature. So now they're actually going to look at this and they're going to have to figure out their evidence reasoning and then make a scientific claim. For lab number 4, we have another different type of graphing assignment with different type of data. This data set is looking at true data that a scientist has given us from Mauna Loa up in Hawaii. And so we have data from 2006 all the way through 2019. Students are not going to be doing all of the graphing. I only put one. This goes across the entire room. So we have whiteboards, and if we go across the room, each group has a set of data, and then when you line them all up, you can see what's going on at the time. And based on that, they can see that the amount of carbon dioxide is definitely increasing over time from 2006 to 2019. Again, we have a prediction in our same format. What will happen if humans continue burning fossil fuels at the current rate? They got to take this data, right? They got to get evidence from all of these maps across the room, or not maps, but graphs across the room. They have to reason. I even give them a hint. What will happen to the sea level, right? So now we're bringing in our background knowledge, and then they have to come up with some sort of a scientific claim for that. The big one that we have is the Anthropocene Ice Core Lab. This one has them, they've learned different skills all along, and this one kind of wraps it all up in a really nice bow, where we have these resin ice cores that my sister made. So here it is, we can pass it around. I know you guys have seen one. You haven't seen one. But so the resin ice cores, inside of it, you can see that there is different types of data. So there is some yellow little beads in there, and then there are some red little beads in there. And then those are going to represent the carbon dioxide. Those are going to represent the CH4, which are greenhouse gases. You can see by the end, there's much, much more of this. And then they have to go through and they have to make these evidence and reasoning and claim type of observations again. And really what I'm looking for now is more in depth than what we started with, which is what's the carbon dioxide reasoning? Why is there more, right? What about the methane particulate matter? And then at the very, very end of this whole unit, we have an assessment, a formal assessment, where I'm going to give them a graph, and then they have to go through, and it's the same format, so they shouldn't be surprised, like, oh, where did this come from? But do you think that humans have caused the current pulse of global warming, right? And they better have some evidence. I even gave them a really, really nice hint based on the data. What evidence is there that shows an increase in carbon dioxide over time? They just look at this. Pretty easy. There's only one piece, like they're only graphing one, so they can't mess it up with the temperature here, just the carbon dioxide data, and then make a claim. Make a claim about the rise in carbon dioxide levels. And they have a little bit of a carrot, a little piece that they can make a scientific claim about if they're kind of lost. So that's the end before we get into taking our actual data. I would like to thank, for my acknowledgement, my mentor, Dr. Janell Webb. I got to sit in on his, what is it, the PhD dissertation? Defense. The defense, okay. So I got to sit in on that, and then it was really interesting. It was during my prep. My kids came in and were like, ooh, this is interesting. So then they all sat down. So we got to all kind of talk about this and how this was going on real time. And Dr. McDonald, he was the principal investigator in this. He's now at UC Berkeley. Adrian Hart, he is a PhD candidate over in the geology department. And Frank and Marilyn for all of your support and pushing me to make really good, clear models. And then, of course, Nicole and Bill for up in the room, just listening and all of your input and ideas and support. So at the very, very end, there is some standards that those are at the end and they're also in the. So
thank you.
There's that.
Any quick questions before we look at some of her I-scores?
Really?
Well, there's so many cool threads of interesting activities you could look at. I was thinking about how you even found one that's like a bracelet that you could use as like little beads, could be possible little data points for climate if you count and look at the colors of the beads. That was cool.
That was
so
cool.
there's a
that would take forever though.
Yeah, there's multiple threads, and you certainly went down a few nice ones
that are very doable and not expensive. And you know, when you think about it, because if you have 150 students to do this. So.
Yes.
Yes, if you have any thought about using settlement cores,
this is ironic that you would say that. So Maryland found these really cool things that are actually in my car right now, but they're push-ups. Like, you know, do you remember when you were little and you had those little push-up ice creams?
Oh yeah.
So she found some of those. So yes, we're thinking about Jello and then having the kids go like this and then pulling it out, or a cake of some sort where you could put it in and then pull it out, and then you can have what you can see all of this. And I could put either seeds in there or little candies or whatever. But yes, I have actually thought about that. And I do have a well-logged lab that that I would definitely do something like that, where the kids all get data that's like for a well log, you know, and then they have to actually draw the thing based on their data. And then at the very, very end, then we put them all up, and then you can see a whole process and what's going on and where the point of detection is with the water.
Yeah. So I want that make them understand more that things happen here too, not just going forward.
Yeah. That would be really cool.
Yes, with this, I really enjoy how you changed up the CRE order, and I love how it's like scaffolded on one side, and I think I'm going to try to use that. So are these the hints you would give your concurrent enrollment students, or are these the hints you would give your chemistry students?
I would give them all.
Or you would give it to both of them. Is there anything you think you would add for the chemistry students?
I haven't taught it yet.
Okay.
So I don't know. But after the first time that I teach it, I would definitely know.
Yes.
So if you're here in like April, I'll try to actually get this in before we do this, because I think it would be very important, because it's saying all the teachers are going to want to know. And so I already have people on Twitter, because I put up my I-scores on Twitter, right? And they're like, oh my gosh, this is awesome, because I have like 1,400 followers. And they're like, oh, let me know how it goes. I really am intrigued here. And so they will probably get this lab, and then they will also give me their feedback. I don't know how it would have to be scaffolded more, but I would definitely be open to scaffolding it.
Yeah.
And Bill was the one that came up with this, because he's like, the claim doesn't make any sense. And then my department chair and I have already had these conversations going around and around and around. Like, why would you do that? And then finally, she just came around.
Teachers a couple weeks ago.
Yeah, so she just came around and she's like, I think I understand why you would do that now. So essentially, the statement that you're giving is the claim that they would normally have to do. But this one, they're making up the claim.
Yeah. And how would you support the neediest of students in? There's nothing in no supports in the claim box yet. Like, how are we going to support?
Well, you know how I
there?
would do that is I always, for the first one, we do the first couple together, right? We do the first, and then so I have my little document camera, and we're like literally doing it, and then we have a conversation. Okay, what do you guys think? And then I will write down something, and then I go, but if you think something else based on your data, you write it down. And by this time, we've already done this thing called observation versus inference. Why did this dam break? But the dam didn't break. There was just flooding down streams. We've already watched this video. And so they have to write down all their observations that they've seen on this video. Now, what do you think happened? Well, the dam broke. Everybody died downstream. And it's like, no, actually, there was a landslide up above the dam that didn't displace the water, that went over the dam and then went. And they're like, how are they supposed to know that? I'm like, oh, you didn't. but now you think about that, right? And so they always do these observations for ceasing things.
Do you teach any EL students?
I teach a lot of EL
that also maybe putting some translation in that.
Yeah, exactly.
I like it from that.
I can see you're gonna try it too.
Yes.
You're gonna do that. So shall we try my little?
Yes.
I mean, now I'm behind.
Being here, my name is Nicole, and the title of my curriculum is Root Harvest, Energy Harness, Exploring Battery Anatomy. You can decide if you think those are rhymes or near rhymes, but I thought it was really catchy. A little bit about me, as Frank mentioned, I am a teacher over at Thatcher. I've been there for 2 years now as a fellow. I was hired right out of undergrad after studying chemistry and education, and I'm one of those nerds who in high school knew she wanted to be a chemistry teacher in high school. So luckily I got the chance to live out my dreams at Thatcher. Under a mentor, I've been teaching integrated science 2 for the past 2 years. So that's our chemistry and biology sequence for 10th graders. And what's really cool about that curriculum is there's a pretty heavy emphasis on environmental justice. Being a private school, we can kind of not have to be wedded to NGSS and explore themes we think are important. And so you'll see throughout this presentation as well, environmental justice does pop up a lot. And then this upcoming year, I've been hired full-time, so I'll also be teaching Integrated Science 1, which is our physics and chemistry component. So that's for 9th graders. So I'll kind of be teaching the whole sequence as a flow. And then on the side, I also do curriculum development and teaching for health education at the school, which is a lot of fun. So to talk a little bit about my RET1 experience from last summer, I was placed in the UN lab, which is focused on photovoltaic devices and organic solar cells. And just to talk a little bit about why solar cells matter and where they sit in the grand scheme of renewable energy. You can see in the top right graph here, even though across the US and across the world, solar cells are only increasing in numbers, their efficiency is still relatively low compared to a lot of other renewable energy sources. So things like nuclear and geothermal are harvesting a lot more energy relative to solar, which sits at about 20% right now. Of course, there's a lot of research to try to get that efficiency up, and that's one of the main focuses of the UN group. The other main focus is trying to use materials that are organic, which are much cheaper, better for the environment, and much more tunable than the silicon counterparts, right? So you think of a solar cell that you see, a big solar panel, typically that's made out of silicon crystals, an inorganic material that's relatively expensive. But the cool thing about organic materials is that you can make them through organic synthesis, and you can also optimize them based on what kind of properties you want them to have. Nevertheless, right, all of these types of solar cells follow the same general pattern. You can see here, you get sunlight exciting electrons in some sort of atom. Here I've used a carbon atom because that is the backbone of our organic molecules. And when that sunlight comes down, we separate an electron from its corresponding hole. So I won't go into that too much because it's very confusing, but essentially an electron in the hole pair is called an exciton. When we separate it, we actually allow charge to flow through the whole contraption. So sunlight allows that exciton to separate. Electrons are able to migrate through this thing called the active layer in the middle, and then be released into the cathode and then be flowing through as current in an external circuit. So that can power things like light bulbs to our cars to whatever kind of scale you can think of. And the cool thing about these types of solar cells is that you'll notice the active layer is not just made up of one material. It actually has two. It's usually called a heterojunction cell for that reason. It actually has donor material and acceptor material. So that actually allows the electrons to flow more efficiently and more smoothly rather than if there was just one of them there. And so my work in the lab last summer was looking at, okay, what types of organic materials are we putting into that active layer and how can we optimize them, make them more stable, able to withstand heat, et cetera, et cetera. One metric you can use to measure that stability is called TG, or glass transition temperature. So this is a really cool property of polymers in particular. And essentially it's the temperature at which a polymer will go from very hard and brittle to very rubbery and flexible. So essentially the higher your TG is, the more amount of energy it will take to get that polymer into a rubbery, flexible kind of decomposing state. So what we're looking for is something with a high TG, so it can withstand lots of heat. So the main research question I was looking at last summer was just thinking about one type of molecule, Cotic 4F, which I won't even try to pronounce the full name of, But this is a non-fullerene electron acceptor. Fullerenes are often studied, they're called buckyballs, right? And there's a lot that's known about them, but things like this that are tunable, that you can make in an organic lab, we don't know as much about their properties. So it was important to try to get some data about that. After many weeks of doing various types of analytical methods, I arrived at a TG of 76.9 degrees Celsius for this particular material. So it was a lot of like trial and error, learning the methods last summer, just to arrive at this one data point. So that might seem like, oh, that's not very much, but it is important if someone ever wanted to look at this material and see, okay, what are its properties? What's the database of research that's been done? Theoretically, this would be one data point we can look at. So what else did I learn last summer besides all about OPVs and solar cells? Kind of thematically, I took a lot away from this experience. First, I just got a lot of experience as a scientist and kind of thinking like an engineer. In undergrad, I really gravitated towards opportunities to teach and be a teaching assistant and be a tutor, but I didn't ever actually work in a lab. And so this was a great chance for me to finally get to do that and also compare what is it like to contribute to standing theories versus try to optimize a solution, right? So that subtle difference between the scientist and the engineer. Secondly, like you all, I have an appreciation for material science now. I love interdisciplinary connections. I love that I work with people who are trained physicists and chemists and biologists and engineers, and we all have this shared goal. And so really kind of trying to bring out those connections more. And then also I got a lot of perspective and education about renewable energy sources. So obviously it's important to be able to understand not just solar energy, but how this compares to other types of renewable energy, which will be increasingly important as we continue to warm up the globe, like Laura said. So what I realized is that I could actually kind of take these thematic buckets and translate them into my curriculum directly. So you'll see, hopefully as I talk, kind of these threads that are woven throughout my curriculum. First of all, trying to get the kids to understand what does it mean to approach a problem like a scientist versus an engineer. So that'll come through in the lab activities. Building a better battery and this idea of, okay, we have this big problem in the world. There's a lot of ways to get renewable energy, and actually, how do we actually store that energy in a useful way and then use it to power something useful? And then thirdly, perspectives on renewable energy. So thinking about that process of transferring energy from one source to another versus actually storing it and saving it for a rainy day when it's going to be useful. In terms of NGSS, like I mentioned at my school, I'm not wedded to these standards, but for any teachers wanting to use this content in their curriculum, I have highlighted a few standards here. So the first one being HS Engineering Design, the second one being HSPS3 Energy, which is a physical science standard, and then another physical science standard being matter and its interactions. So these latter two kind of fit more into the chemistry and physics vein, but the engineering components can kind of go into any form of science class. And as you can see, that kind of aligns nicely with those sort of qualitative themes I was gathering from my experience last summer. Okay, so now getting into the curriculum, I'll mention for my school, I was only able to really find space to incorporate this into 4 days. At Thatcher, we had 70-minute classes that only meet 2 to 3 times a week. So that would end up spreading over 2 weeks. And that would also incorporate 3 to 4 nights of homework. For another teacher, that might look like 45-minute blocks more often, so it could be tweaked. But the main phenomenon explored in this curriculum is really, what does a battery do? How does it work at that atomic level? And why do we care? Why should we care about how this works and how to optimize them? So it kind of aligned things along the 5 E's of guided inquiry model of science education, but I will get right into it. Okay, so the first day, the students will be really engaging with this idea of what is a battery and what are some different models that a battery can look like. So I have one of them set up right here, which is a sea salt battery. I had some minerals from the Dakota coast of Hawaii, which contains like magnesium and potassium and sodium, and I dropped them into some water, and this battery actually conducts 0.5 volt, so not very much. But I'll have this set up next to a potato clock. So something that is basically 2 potatoes with some electrodes and then powering a little clock. And ask the students to compare, how are these things similar? What do you notice about the materials used and what is different? So really not necessarily giving away all the answers yet, but just having them think through that phenomenon. And in addition, students will be looking at some redox reaction slides. So I'll kind of go through that core content of path reactions, oxidation states, how are electrons transferred in these reactions, and give them that vocabulary to understand what's going on. And I should mention, there's a big component of this is just the teacher slide deck. So that's really anchoring all of these lessons through and through. And there is a good amount of lecturing that will need to be done in accompaniment with these lessons. So the homework for this, students will just go through oxidation states, thinking about how those are assigned, really practicing that skill. And they'll also start to think about energy transfer and storage, researching some non-renewable and versus renewable sources, and thinking about what does it mean to harvest energy versus capture it. On the second day, we'll have our first of 2 lab activities. So this will be a little hands-on experiment with making 6 different voltaic cells on one apparatus. You can kind of see a picture of it right here. It's basically just a piece of filter paper with 6 little petals, and you put a metal on each petal, no rhyming intended, and try to see the conductivity of each one compared. So this really allows students to assess what is the conductivity of different types of metals relative to each other, and then compare that data to the standard reduction potentials table, which is a key concept in chemistry. So for this one, sort of like Laura said, CER is also a struggle for my students. And so I would be kind of giving them the hypothesis, right? The voltage that your battery can produce is dependent on what your metal's identities are, but they're going to have to do the hard work of finding that evidence and reasoning to support that claim. So for homework, they'll work on a write-up, and then they will also watch a video on the extraction of lithium and copper and cobalt, just to kind of start zooming out and seeing what are the environmental ramifications of how these materials are actually harvested, right? They don't come out of nowhere. They come out of a lot of hard and toxic wood. On the third day, after they've kind of turned in that lab report, we'll kind of bring everything together and consider what does the battery actually have going on in it. We'll talk about this phenomenon of charging a battery versus discharging it. Like, what does it mean when you charge your phone battery? What does it mean when your battery dies, right? And kind of using lithium ion batteries as a case study in that. The Clermont lab had a really cool video of that I might show there. And then also kind of zooming out to have a broader discussion of building a better battery doesn't just mean how do I make this bigger and more useful for society, right? It also means how do I do this sustainably and equitably and treating Indigenous community and land with justice, right? Which is a huge issue right now, especially for cobalt in the DRC. So in class, we'll start to talk about those ideas, and then we will also introduce the fun fruit apparatus project. So here, students will kind of use all of the content they've been learning about batteries to engineer their own design for a fruit battery and try to get as much voltage as possible out of it. So I have here lots of different types of LED lights ranging from like 2 to 3.5 volts. So they can try to make their battery as conductive as possible. And here they're kind of shifting from a scientific mode to more of an engineering design mode, right? Like the goal is to optimize this, but I'm not going to give them a step-by-step procedure of how to accomplish it, right? I'll tell them, here's the materials you have, here's the background knowledge you have, how can you approach this to optimize the solution? So they're still kind of practicing CER, they're practicing picking variables that they want to tweak. They could say, I want to vary the metal identity, I want to pick different number of fruits, I want to pick fruits and potatoes, right? Comparing different types of materials. So they're kind of still practicing picking variables and testing them, but still from more of an engineer's perspective. So for homework on that day, they'll work on the design, and then on the final day of the class, this is sort of the summative assessment here, they will actually construct their fruit battery in their groups or pairings. And so they'll test it out, attempt to maximize the voltage of it, and then kind of go around the room, do a little gallery walk and see what are other students' batteries looking like, how did they meet success in their designs, and what does that really mean in the context of engineering? And so here they're kind of getting that evaluate component, and they also can do the post lab in class if there's time for it, or it could be assigned as homework depending on how the curriculum flows. In terms of how I will assess this kind of pilot of my program, my current chemistry curriculum has a gaping hole for redox chemistry, so I had to create all the slides for the content as well. But this will come at the very end of my term, so kind of be a nice conclusion to our chemistry year. Two ways I thought of kind of assessing their learning. First of all, they could complete this pre and post reflection. So before we even start the unit, just asking these 4 questions. How does a battery work? What do you know about this already? What kinds of processes in your life are used by batteries? And then having them answer the exact same questions following the unit, just to get a concrete measurement of like, what did you actually learn in these 4 days? And then I have here hyperlinked, but I also have a regular unit reflection I like to do just to get a sense of what questions do you still have about this topic? How is the pacing for you for this unit? So was it too fast, too slow, just right? What's the most beneficial activity for your learning? Was it a lab? Was it a homework assignment? And what was the least beneficial? So something like that too, to get a sense pedagogically of how it went. Before I let you loose to try some fruit batteries, I just have some thank yous in order. Dr. Janell Webb, who has since graduated and in Korea, the whole UN lab for having me last summer. I learned a lot from the experience. Frank and Marilyn for all of your support and your patience. This was my first time actually building curriculum, and it was not easy, so it really helped me out. Laura and Bill, you are amazing mentors. You are inspiring to me, and I appreciate you guys so much. And the UCSB and MRL for allowing me to be here, and my roommates for being great supports along the way. Donnie for setting me up with them. Thank you all so much. So Frank has nicely printed out part of the curriculum here, but basically there are some like pre-thinking questions that students will think about before they design anything, and then during class on that last day, they'll actually put their idea to the test. And so I'm happy to help you out with any sort of things, but these all came fresh from Thatcher campus. So yeah, you can also eat one if you want. But I have potatoes here, I have citrus fruits, I have zinc and copper. And in case you don't know anything about a battery, the electrons flow from the zinc to the copper. So what you do is essentially try to build a series and circuit type of thing, and I can help you out with how to wire stuff. This is all like in the teacher slide deck as well. So I would be able to teach students like, here's actually how you hook up things to make them flow correctly. And then also here's how a digital voltmeter works, because it's important to be able to put the leads in the right place and things like that. So all of that is kind of in the teacher.
Awesome. Let's get into it. Any quick questions on anything else before we dig into the fruit? The salt bridges, I should say. Awesome.
Janell, as you mentioned, Janell Webb. And my project this summer was identifying key factors that drive DNA methylation reprogramming of human CLCs, which are primordial germ cell-like cells. So this is my first summer here. So this is my RET July program, and then you can hear from RET students. So my mentor was Zovia Siglitz, and she is a PhD student. She just finished her second year here at UCSB. My faculty mentor was Siddharth Day, and our funding was through the National Institutes of Health. So I will be teaching ecology and serving as an academic dean at Aspen Christian Academy in Washington. We'll maybe on Sunday. So I'm enjoying my trek. Let's take off. I have 3 kids, 6 grandkids. They all live up in the Washington area. That's another reason I'm in that direction. I love hiking. I love spending time outdoors, especially with my pups. I have degrees in animal health, biology, and public health. And so having studied animals, people, and nature, I am absolutely fascinated by the interconnection between the 3 and how each one influences the other. And I tried to bring those connections out of my classroom when possible. So our research goal for this project was to identify key factors that drive DNA methylation reprogramming of human PGCLCs during early human embryonic development. So primordial germ cells are the specialized embryonic cells that are formed early in the development of the embryo. And in the diagram here, there's the green cells here, and you can see that they migrate to the gonads. The PGCs are the precursor, they're the germ cells that do meiosis to form the egg sperm. They're the only cells in the human body that do meiosis. DNA methylation is the addition of a methyl group to the nucleotide cytosine on the DNA, and it generally occurs in a GC pair. So here we have a cytosine, and the methyl group is attached to the first carbon. DNA methylation is associated with long-term transcriptional repression. So here, if it is methylated, transcription doesn't happen. We don't make proteins from this particular gene. Here, in an unmethylated version of that same gene, then you will get transcription and you will get your proteins. This methylation is passed on through mitosis, so it seems to be a memory mechanism that helps to differentiate cells, because the types of protein cells may differ. And so if they don't make certain proteins, that helps to determine, to differentiate those cells from other cells. So if you remember for your central dogma from high school biology class, right, DNA is transcribed into RNA, and RNA is translated into proteins. Proteins are the regulators of other tissues and organs. They're structural units. Each one has a specific structure and operates in a specific environment, and therefore has a very specific function. So in Sophia's project, we were using omics techniques to study the central dogma. So omics techniques analyze biological molecules comprehensively on a large scale. So omics is large data sets, and it really came about as a necessity from the Human Genome Project. So there was massive amounts of data being produced as we were decoding the human genome, and there wasn't a good way to analyze it. So omics techniques kind of developed out of that. So it's the analysis of huge data sets to get a comprehensive look at how molecules work in systems. So combining multiple omics gives us a clearer picture of how molecules are behaving and how systems are working. Sophia and I spent most of our time looking at transcriptomics, which is the RNA sequencing, and looking at gene expression in different cells, and then also in the epigenomics, the modification that we do in methylation. So 1 out of 6 people worldwide struggles with infertility, and currently solutions don't always work. They're very expensive. They're also very physically demanding. So the insights gained into epigenetic dynamics of human PGC development could allow for the synthetic production of the proteins from them, which could be a possible solution for infertility in humans, but could also have applications in animals, species regeneration. Obviously, there could be and need to be some ethical discussions about synthetically produced proteins because it could really change our understanding of human organs. If we can make the proteins without parents from a stem cell, then that kind of changes how life begins. So our methods that we employed over the last 4 weeks included lots of lengthy protocols, basically following instructions, following recipes, and also using many interesting machinery. And one of the things that I found most fascinating about this machinery is that it all took advantage of the types of molecules and the structure of the molecules that we were trying to understand. The workflow for Sophia's project includes, first of all, creating the primordial germ cell-like cells. And she did this before I arrived, at least the cells that we were working on while I was here. And she goes through a process to create those chemistry, makes biology happen. In an argument between myself and my husband who teaches chemistry, he wins because chemistry makes biology work. So this is the chemistry of creating the PGCLCs. Okay, here we're using CRISPR guide libraries to add plasmids of specific genes to the PGCLCs to understand what those genes do in the creation then of the agonist sperm from the PBCL seeds. The flow sorter sorts out the cells that have the plasmids in them. They're tagged with GFP, the glowing protein from jellyfish. So then when you sort them, they glow, and then you can pick out the ones that have your plasmid, and you can sort them into single cell wells so that each cell can be studied individually. And then the sequencing identifies the nucleotide sequence in the DNA, the actual sequence of what we produce. So I spent most of my time working on the sequencing aspect, preparing the DNA samples for the sequence. But we also did a little bit of this plasmid cloning to prepare for the next round of PBCLC studies that should be doing. So cloning a plasmid or making a plasmid just requires that you take a piece of DNA that is of interest and snip that out, and then you get a plasmid, which is a circular piece of DNA that is produced by bacteria. You snip out a section of that, and then you put the DNA you want into the plasmid, and then you can send the plasmid into a cell through a vector. And so here is using lentivector or lentivirus, which is actually a modified version of virus. To implant then what you want, that gene, into your cell. So this is the plasmid that Zoe created. This is the backbone of the plasmid, and she used CRISPR to do this. She designs it on a computer, and then she orders it. And somebody else assembles all these pieces and sends them back to her. And then she uses this backbone then to as this part that she then snips a piece out, and it's the ESP3I here where she cuts it and then inserts the genes that she wants to study, which can then be inserted into the cells. So to clone plasmids, we already have these plasmids. She had already made them. We snipped in the piece that we wanted, but then to grow more of them, you grow them in bacteria. So here we have a cell plate where we grew the bacteria. We got really good growth. There's lots and lots of colonies on there. And then we can take those colonies and we can do PCR. So increasing the amount of DNA through human reaction. And then the nanodrop machine, we can figure out the concentration of how much DNA we have to make sure that we have enough to do with gel electrophoresis. And the gel electrophoresis then just sorts the fragments that we got from PCR to show us if it did in fact digest the way that we thought it should. So if it's the correct plasmid, if the plasmid cloned correctly, then we should see these things. So these 3 clones were successful, and this is the best that we ran through without any. So that worked to create clones. And so then she now has a supply of plasmids that she can use in the future. So I spent some time doing cloning. Also spent a lot of time preparing DNA for sequencing. So dyad sequencing is used to understand the link between DNA methylation and the single cell identity. And the Day Lab recently developed a brand new method to jointly analyze DNA methylation and the transcriptome. So DNA methylation is the locations on the DNA where the methylation has occurred on the cytosine, and then the transcriptome is the parts of the DNA that are being read into RNA and then protein. So this paper was just published last April, and I had the privilege of working with Samia and Liz and Sid in the lab. Alex has already graduated, and Shara was from UCLA. So this process, in this process, remember DNA methylation is the attachment of the methyl group here to the cytosine, and in the GC pairs. And this MSPJI restriction enzyme that recognizes locations along the DNA where one side is methylated and the other side is not. And restriction enzymes snip. Enzymes are molecular scissors. So they snip these fragments into sections whenever it recognizes those locations. And then after the conversion here, those methylated cytosines will not be converted because they're protected by that methyl group. And so they continue to be C's in the end. So unmethylated will go from C to G and then G to C again in the sequencer. But the unmethylated ones will convert to U's, which then convert to T's in the final. So when we compare those sequences back to our reference genome, which is just the template that we used to start with, we get these locations, which then tells us where the methylation occurred. And so in the end, what we're producing is a library of all the methylated GC locations on that DNA. And because DNA methylation is an important factor in creating those PDCs and then those egg and sperm cells, understanding that map would then give us the keys to create those cells ourselves. So this protocol is a 10-day process. I'm not going to go through the whole thing for you, but I did want to talk about the bead cleanup and give you a little bit more information about that because I found it absolutely fascinating. So the bead cleanup uses SPRI beads, and SPRI stands for solid phase reversible immobilization beads. And they're made of a polystyrene core, and it's surrounded by magnetite. And then that magnetite is coated with carboxylate groups. And they're paramagnetic, so they're magnetic only in a magnetic field. And they're used to purify DNA samples. So they take advantage of the fact that DNA is a negatively charged molecule, and the negative charge on the DNA is attracted to the carboxyl. And then in the presence of a crowding agent, and then that's a polyethylene glycol in sodium chloride solution. And so the DNA will stick to the beads, it's attracted to the beads, which then allows you to remove any debris from the liquid that is in your tube. So this is the protocol that we use. We begin with a PCR mixture because later once you have your purified DNA, you're going to replicate and amplify it with PCR. So we add a PCR mixture and then we add the beads, and then we wait about 10 minutes, the beads bind to the DNA, giving a chance for that negative charge to be attracted. And then in the presence of a magnet, the beads will stick to the side of the tube, and that leaves all of your debris in the liquid. So you can draw the liquid off and you have just your DNA in the tube. We wash it a couple of times with ethanol just to make sure we got all the debris out of the tube. And then we can release the DNA from the beads with nucleus-free water. So water that has no nucleus. And the water just causes the beads to let go of the DNA. And now you have your purified DNA in the liquid. Your beads will stay up against your magnet so that you can withdraw or you can purify DNA. So this is what that looks like. This is a video of that process. So this is a magnet with a test tube holder. And you can see that the beads stick to the magnet and the liquid just starts to become clear. So when it becomes completely clear, you can just lean the magnet back and the beads will stay on the magnet and you can draw the liquid out. So I believe this one was still, we were drawing off the debris and washing it. And then in the end, you're drawing off your purified DNA. So results of what I was able to do in the last 4 weeks are not quite complete because the samples that we prepared have to be sent out to be sequenced. That takes about 2 weeks, and then we send out later this week. So I might get to see some of those later if they do send them to me. But before the sequences, before any of the samples are sent out to be sequenced, we do check them with a bioanalyzer to make sure that they're a good sample, that they have enough they have a high enough concentration really to be sequenced properly. So we were able to use the bioanalyzer on our samples just this week. And the bioanalyzer uses a chip, and it basically does gel electrophoresis in capillary tubes. So you're using 1 times 10 to the 6th less sample than you would if you were running an entire jet. And because we're working with very purified maybe I want to make sure that we're not using a lot of it to check before we send it to the sequence. So this bioanalyzer shows us if the marker genes are being expressed. It also then shows the protocol effective, and it shows you the concentration of the sample. So here we can see we did plate 1 here. We did 4 different samples from plate 1, and they all have this gene right here, which is what we were looking for to make sure that our protocol is good. And then in plate 3, or sorry, plate 6, these 4 samples, you can see this gene was obvious, and that's what we were looking for. So this shows us that our genes are being expressed and our protocol seems to be effective. So then these graphs show us concentration. So in this sample, which is plate 6-1, this is a lower marker and a higher marker, and what we want is this nice kind of hill in between, where this is not a lot higher than this and falls nicely before it gets here. This one you notice is now in top. And this is showing me fluorescent units. So how many of how much am I doing is fluorescent? And then this is showing me base pairs. So here we have approximately the same base pairs. So we're looking at, again, the same DNA and the same genes, but we don't have good concentration on it because we don't have a lot of fluorescent units. So this sample probably won't be set. It needs to be PCR to make sure we have enough concentration. We don't want to send it if it's not. It takes a lot of money and a lot of time to sample. So we want to re-prepare this one before we send it at this point. So even though we don't have I don't have all of the data from the sequencing, we do know that the samples that we produced, most of them are good and are going to be sent to the sequence. And then from there, she can build her methylome library, the library with all the methylated sites, and she can start going through the encryptome library, which is the sections of the DNA that are derived into specific. So my overall takeaways from my summer in RET that I personally learned from this past 4 weeks is a details measure. That following instructions and carefully reporting information is absolutely critical to experimentation. All those little tubes and switching from one tube to the next tube to the next tube after everything very quickly. Also, that experimentation requires a lot of time. We did a lot of check and recheck. We did a lot of the same thing over and over again to make sure that they were done correctly. Also, understanding the structure and function of molecules allows us to find ways to manipulate them and learn more. So I emphasize structure and function all the time in my section. Structure defines function. If you know function, you can figure out structure and how those store together. And I thought it was absolutely fascinating that so many of the protocols and machinery that we use took advantage of structure, and specifically the structure of the molecules to manipulate them. And then I also learned that lab skills improve with smart pipetting skills improved greatly over the last 4 weeks after pipetting every day. So how could I take this to my classroom then? Well, I definitely would like to provide more opportunities for my students to follow instructions, carefully record information, something that they struggle with, and I realize that the more that they get to practice on, the better they can get at it. Also, I can now add more real-world applications to my emphasis on structure and function because I have a number of them now. I also realized that if I want my students' lab skills to improve, I have to give them opportunities. So I get frustrated with them because they don't remember how to clean a test tube, but I only let them clean the test tube once every 6 weeks, 4 weeks. So if I want them to learn how to do those things correctly, the kind of material structures, then I should let them practice. So possible curriculum connections that could be made. There's obviously a lot of science skills here that we can pull, following scientific protocols, carefully recording data, petting and testing, examining materials correctly. Also using math skills. And one was I realized that my math skills were a little weak and had to work on that a little bit. calculating percents of the formulas and how much of each thing we need to have. Also, lots of easy genetics unit connections, DNA, RNA, molecular structure, plasmid structure, function, cloning of plasmids, the process of bacterial transformation and cloning of cells, the gel electrophoresis procedures and purposes, as well as science of omics and bioinformatics. I love to present career opportunities to my students and help them understand that science isn't just research, but there's also, if you love computers and science, then you can put those together in bioinformatics. If you love art and science, there are avenues that you can use to use both of those. But I will not maybe be teaching genetics. I'm not going to be teaching cellular biology next year, and I'm not sure about the year after, but next year I'll be teaching ecology. So thinking about ecology connections was a little bit trickier. But because the cloning of cells and the creation of the synthetic plated sperm could be used for endangered species, this is something that I think would be actually considered conservation biology when we're talking about endangered species and solutions for the protection and regeneration of species. It also could fall under natural selection and artificial selection, thinking about how nature preserves things that are beneficial, and then how we as humans interfere or assist in those areas. Cycles in nature also involve a lot of single-celled organisms, so because we were working on a cellular level, and also with bacterial transformation that can possibly come into cycles in nature and thinking about how bacteria participate in the carbon cycle or the nitrogen cycle. And then genetically modified organisms is a big issue with food sources, agriculture, and feed animals. And so this could also be something that we pull in with the cloning and the plasmids and the cellular level of change and DNA modification. So these were some possible things that we kind of brainstormed a little bit yesterday as options for pulling this information into college. So it was a great summer, and I just want to extend my thanks to Sophia. She was an incredible mentor. She was really patient with her time. Dr. Sid Cosplay and all the members of the data research team welcomed me into my lab. Everyone was very friendly and helpful. Also, Frank and Marilyn for their support and guidance. And then also, Ian and Casey are not here. They're my RET1 cohort partners. And our meetings each week are really encouraging and very great feedback. And I enjoyed working with them this year and look forward to working with them again next year on our RET programs and writing this amazing curriculum that I have heard from the RET groups. So thank you for listening, and are there any questions?
I have a question. How do they snip the little pieces?
That's the restriction enzymes. So all restriction enzymes have specific codes that they recognize. And so if you use a restriction, and she was using ESP2 or ESP3, that ESP3 recognizes that mutation on the plasmid and makes the snip.
So some big giant machine.
Well,
Not like little pieces that are so small.
it's an enzyme, so it's chemical snip.
Oh,
So
okay.
yeah, so this ESP31, it recognizes a specific location. It has a specific code that it recognizes, and anytime it sees that code, it causes a break in the bonds within the DNA. Yeah.
I thought they were little.
No, they aren't actual. Yeah, it's all done with restrictions. It's chemistry. So I call it chemistry, it's biology, because chemistry and biology. So yeah, restriction enzymes are fascinating, and all of these little cells, when she does this, she puts in a barcode. There's an 8-letter code, a different code for each of her cells, a little barcode on this, so she knows which plasmid is in which cell when she reads the DNA. Let's get a barcode in there. So we'll be barcode, and so yeah, there's a how do you get a barcode? It's just a DNA code. It's just 8 letters, and it's unique to that cell. Which I just think that's it. And we're talking such tiny molecular level. And all of this is done by chemistry, and then you check the recheck to see that the different stuff. So it's not impressive.
These results that are on the way, or whenever you send it in, will Sophia and you be looking for the presence or absence of methylation sites, or are you looking for things to turn on or off? Or
she's looking at methylation sites, and she in these plasmids, she's inserting different genes, or she's increasing or overexpressing some genes and underexpressing some genes to see how that affects the methylation map. So she's looking at where are those methylations increasing or decreasing in specific cells compared to the normal cell. And
all those results will hopefully accumulate into where you know exactly what to do to get back to the germ cell. Okay, and that in your opinion, or is that is that a decade away, or years, or just it's hard to say?
I don't really know how far they are in their actual methylation map. Now they have done this in mice, and so that's proof of concept.
Cool.
They've gone all the way to creating the synthetic egg and sperm, combining them and creating mice from synthetic egg and sperm. So that was proof of concept, but in repeating that, they're finding that the demethylation patterns are different, and so that's kind of where they're trying to figure out how is it different in the human cell and why is it different in the human cell when mice are modeling systems from mammals. So.
I did this sort of thing 20 years ago, and sounds like you've been in this field for a while. Do you have any previous experience with this? And if you do, what have you noticed that's changed the most in the past 10, 20 years?
Yeah, I don't have specific experience on the DNA level. I've taught biology for over 20 years, so I'm pretty familiar with the whole DNA, RNA, central dogma, all of that. And I was able to do an internship my second year in college at the Cincinnati Zoo while I worked on the animal health degree, and they were doing frozen embryo studies. So if this situation wasn't with DNA, but they were harvesting embryos in Africa from endangered species, and bringing them back to the States. The one that made the daughter was kind of into a cow. So the cow gave birth to a rare form of antidote. And so this was, again, it was an endangered species kind of research, but we weren't dealing on the cellular level, we were doing on the embryo level. But that's really the only experience that I had.
I remember doing a lot of PCR training.
Yes.
The last slide you shoot with bioanalyzer, that looks new to me.
Yes,
I don't
that's fascinating. It's just a little chip, and it's fascinating when you use such a tiny sample to get a full gel of results without having to run and use everything at once. So, yeah, the machine and the nanodrop machine, the robot dispenses very, very small amounts of liquid into 365 wells in a matter. Well, first of all, you can type like that small, and then to do 360 wells. That's definitely, like you did a little training on ergonomic, ergonomic use of a pipette, so you don't get stiff done. So, the machinery, I'm sure, has.
Thanks again. That was fantastic.
So good morning. Good to see you. I know you all. So thanks for being here and listening again and hearing kind of the final results of our work this summer. I think most of you know me well, but a couple things that I'm interested in beyond teaching is I'm an avid Dodgers fan, as is my wife. Love being outdoors, camping, kayaking, hiking, laying in a hammock and reading a book. But when the weather doesn't cooperate, I also enjoy playing board games. And so in terms of my educational background, though, I've been teaching for over 30 years and taught mostly science and math-related classes, predominantly chemistry, physics, and biology, coached several sports, mostly basketball, and had some other administrative responsibilities as well through the years. I grew up in Indiana and taught for about 7 years in Indiana in public schools, and then in Indonesia for 12 years in the international school, and then mostly in California since we came back from Indonesia with a few side jobs in Indiana in the middle of that. I have been working at Oaks Christian just up the road in Westlake Village, but this summer, in fact, in 2 days, we're going to be hitting the road to drive up to Washington. We'll be working in Cascade Christian schools. And one of the big reasons, well, there's actually 6 of them, these hooligans, that we're excited to go live there. Instead of seeing our grandkids once or twice a year, we'll get to see them twice a year. So we all live in that area. The last summer in the Scott Shell Lab and in conjunction with Jonah and Shay's lab group, I was able to work with my mentor, Stan Lobo. And the water group, as many of them refer to themselves, always is studying how water behaves around proteins and other chemicals versus just studying those other chemicals. And so I was able to jump in with this kind of a side project with Sam to figure out, well, what might we do with analyzing water to try to understand if hydrophobicity of the spike protein and the COVID virus might have led to certain variants being more infectious than others. And so that was kind of the big picture of our research. And so just to kind of sum it up, hopefully pretty briefly, these little popsicle sticks here on the virus are those spike proteins, and the Pac-Mans are the ACE receptors in the cells of the human, and they have to come together in order for that COVID virus to be taken in and infect the cells. And this is this picture is one of the tools that I used last year and will be using with my students this year. It shows some of the variability of how you can see the spherical shape of the atoms that make up the spike protein, or the ribbon cartoon style shape of the ACE2 receptor protein, and how those things have to come together in this region called the binding site. But for any proteins to come together and bind, because they exist in cells in a watery aqueous environment, they have to free this area where they're binding that you see here in purple. So before they're bound, they would have these surfaces covered in water, and they have to push the water away so that those surfaces can come in contact and bind. And so again, this group that I've been working with studies how water behaves, not how the proteins or amino acids behave. And typically, if you have pure water, there's always between any 3 water molecules an angle of about 109.5 degrees. You can see that represented a couple of different ways. And our primary thought process was, how do we analyze these water triplets and measure this angle when it's not pure water? So an example would be taking a small sequence of amino acids and studying the waters. Here it's just showing the oxygens, just for visual simplicity, and saying, well, what about these 3, this triplet? What's the angle between those? Is it 109.5, or is it something bigger, something smaller? And then how about this one, and how about that one, and this one, and that one? But obviously, you can't measure those with a protractor and a ruler because they're submicroscopic. And so we use computational analysis. And using one of the supercomputer clusters here on campus, we would send it data and have it run this simulation. So the green is a sample protein, and then the little dots are all the water molecules around it as they're moving and interacting and adjusting based upon repulsions and attractions and hydrophobic and hydrophilic relationships. And we had the supercomputer measure just a few molecules deep all around the protein, specifically around the spike protein for COVID. We took that thousands and thousands of data points, ran it through different kinds of code to create these pictures in the same program that I just showed you the animation. So it's a very powerful program. And I showed you some pictures, I showed you an animation, and now I'm showing you how we could color these molecules. And those are some of the skills that my students will be learning as they talk about this curriculum. But what we noticed in our research last summer was, if you look at this, and this is the spike protein for COVID, the original ancestral version, and then the Omicron variant, and looking at it kind of into the end of the protein where it is going to bind with the ACE receptor. And you can see here we've got this kind of dark blue and lighter pink, and then over here that dark blue is now white, and the lighter pink is becoming darker pink, so it's trending towards hydrophobic if we look at the scale on the bottom. And this bottom part of this groove where they connect together, we also see that we've got this kind of light blue area that becomes pretty dark red, again, trending towards hydrophobic. And so we couldn't really draw any formal conclusions at the point when I finished the research, but it did seem to show that there was a possibility that that extra hydrophobicity in the binding area for Omicron could have made it more infectious. So as I look ahead to this coming year and how I incorporate what I learned last summer into good curriculum for my high school students, I obviously thought of a lot of the amino acids and proteins and the geometry and attractions and repulsions and what causes those. But I also thought about the tools and skills that I learned to use and how I can help my students gain those skills and learn how to apply those tools. So I had never used really any of these to any great length prior to last summer. But I did a ton of coding, and I won't probably have my students using a lot of coding for this project that I'll be sharing, but there are certain areas where they'll be sort of coding adjacent, where when they put things into the Bible program, for instance, it's actually kind of little keyword codes. And I was also pleasantly surprised to see there were a number of NGSS standards and also science and engineering practices that fell right in line with the curriculum that I was designing. So the curriculum title that I've come up with is We Want to Pump You Up, the Science of Proteins. And I'll definitely show them the slide and probably show them a little 30-second clip of when Arnold Schwarzenegger visited the twins in their pumping up ceremony on Saturday Night Live back in the 80s, give them a flavor of the 80s, and kind of introduce them to the science of proteins. It'll be with 10th and 11th grade students. It'll be about a 10-day process, I think, with 45-minute classes. That's at least what I think it will require. It'll be during second semester as part of an organic and biochemistry unit. And I teach with a modeling approach. So we'll be using physical models you see on the table, and I'll be mentioning some of these later, but also computer models that you've seen that I learned last summer. And so modeling approach is trying to help them think about a phenomenon, build models, and change and perfect those models as they gain more and more information. So the unit flow, I'll take one day to just talk about the research I did at UCSB, give them a quick overview. Then we'll have one day where they start to ask some questions, and I introduce the phenomenon of how do proteins help in muscle building? And then we'll take a day to build some marshmallow models, which you can see examples of here. We'll take another day to build some traditional molecular models, and then figure out how those amino acids will bond together. I'll introduce them to an AI supported app called Fold It, where they learn how proteins fold. And then we'll actually test a bunch of different protein containing materials to see which ones contain the most protein to answer some questions. And there will be a culminating process of student presentations for this. So this would be the slide that I'll show when we have our kind of question and phenomenon introduction day. And say, okay, as you look at the elements of this collage, what are some things that stand out to you? Or have you eaten any of these items? Do you like to eat any of these items? Why do you like them? Why do you not like them? And what questions do you have about them? And which ones do you think might help you build muscle? And just kind of have a conversation, let them ask some questions. And as part of that, I'll have them hopefully come to, as a group, these two questions as being kind of a focus for us. Well, what makes one kind of protein different from another? Why is whey protein different than plant protein? And why might this protein from a steak be different from proteins of legumes? And try to figure some of those things out. And then which sources for building muscle would have the most protein? There's another question we'll try to address, especially in the culminating activity. I'll also, during that day, have them begin the KWL activity of writing down, what do you know about proteins and muscle building and amino acids? And then what do you want to know about those things? And then we'll save the third column of what they've learned for the end of the day. One of the first activities we'll do after we sort of lay out the phenomenon is having them build these marshmallow models. There's a picture of one with all small marshmallows. This week I tried, it looks like I've lost a couple that's falling apart here. That built them a couple days ago, but I tried some with the large marshmallows for carbons. You guys all had a chance to try that version. And I kind of like they're slightly more able to show some of the geometry. So I kind of like that. I'll probably incorporate that. But they'll each choose one amino acid and build a model using marshmallows. And then they'll look up that amino acid on PyMol, that same program I introduced before with the different visualizations, and see if their model looks like what PyMol shows, and also start to visualize that 3D arrangement better. They'll have a choice activity as well, where they can write a poem or a song or a dad joke or make a meme or write a wanted poster about their amino acid of choice. And each student will choose their own amino acid after doing just a little bit of research, but there will only be allowed 2 students per amino acid, so we have a wide variety of acids. When they visualize it on PyMol, they can see it as stick form or ball and stick form, and they will have used PyMol prior in the school year, so they'll be somewhat familiar, but there will be a few new tools that I'll be teaching them in terms of how you can see some of the organic characteristics in PyMol as well. So this is alanine, for example. So the next day, we'll go into the name is bond, peptide bond activity, and that's where they'll use the traditional molecule model kits. And I have 3 just kind of base amino acids with the acid and the amino groups on the central carbon. And then I have a few different samples of R groups. And so they would have to build all of this on their own from the amino acid that they chose the day before. And then they'll partner with 3 or 4 other students and then run through dehydration simulation to show dehydration synthesis, how the amino acids connect, form the peptide bond. And they'll have to write those reaction equations into their journal. They'll have to draw the diagrams. And then once they've made their 3 or 4 amino acid long peptide chain, they'll get with another group and put their chains side by side and try to see if they can predict anywhere where there might be some hydrophilic or hydrophobic areas and what kind of connections or attractions might there be between the 2 polypeptides that they've created. They'll also then take their sequence of 3 or 4 amino acids and put it into AlphaFold 3. And one of the things before we get to AlphaFold 3 that they will be doing is they'll have to create some sort of a visual display of how this peptide bond formation goes. And so I just found this GIF online as an example. They could create a GIF, they could create a stop motion video, they could just do a sequence of slides in a PowerPoint, claymation, whatever they decide to do, but somehow they need to show this process as part of their final presentation. But back to the AlphaFold, once they've got their sequence of 3 or 4 amino acids chained together, AlphaFold is an AI developed program, and it's at level 3 now. And actually the Foldit program, which I've mentioned, I'm going to show more detail in a second, actually was one of the precursors to AlphaFold and helped them gather a lot of data to build this AI. So it's kind of interesting, the connections there. But they will submit their sequence on AlphaFold, like an alanine, valine, glycine, histidine, for example, and then that will tell them 4 or 5 different probabilities of what the AI thinks that sequence of amino acids would look like when they're connected, what shape it would take. And so they'll put those predictions from AlphaFold 3 into Primol again and see which ones, they think they might predict. Do they agree with this one more than that one, or there's certain reasons why they think Model 3 is better than Model 5. And so they'll be using that AI and also the Primol visualization. All right, and then the next day, like I said, Foldit, I mentioned a little bit ago, now I'll tell you a little bit more about it. It was a precursor, like I said, to AlphaFold 3. And they'll be using this one, and I'll just demonstrate it here briefly. When they open up the program, they'll go to this campaign mode. And the campaign mode has, oh, sorry. We got to exit out of this. Main menu. They'll open up this main menu here and go to the campaign mode. And then on the campaign mode, there are many, many tutorials. I won't have them do all of them as required work, but they'll do the first 12. And most of them just take a minute or less. Some of them are 2 or 3 minutes maybe, but they're all pretty short. And so when they open up this program, it's going to be like a tutorial. So it says, welcome to Foldit. And then it talks about clicking this when you need help. And then it says you're going to be dragging these side chains with your mouse. And then it says you want to know about side chains. And in the handout that they're given, they have questions. And so the questions are going to be answered by them reading through the information that they wrote. And some of them are just pretty much directly what they're being asked and shown here. And some are extension to see if they can make some predictions and think a little bit outside of the box. So here it talks about the side chains and how there's 20 different species and thinking through, oh, wait, there's 20 essential amino acids and trying to make those connections. And it just slowly walks them through. It talks about clashes. And so one of my questions is, well, why would there be a clash? Why would some of the atoms being close together cause a clash? What have you learned so far that might cause that is one of the questions on their handout. And it talks a little bit about them and how it's visualized in this animation. And then it tells them they're supposed to get rid of this clash to win this phase. Again, reminds them of where they can get some help. And then they just do it. They move it. It says, great job, you did it. And then after that's over, they can choose the next puzzle and it starts showing the next level. Okay, now let's talk about this. And it just keeps branching out and teaching them a lot of important information, vocabulary, concepts that are relative to why proteins fold the way they fold. So they'll spend one day going through some of those tutorials. And then the next day we'll begin with a group activity. If we had time, I'd do it together with this group. We need probably a few more people, but ideally, if you have close to 20 people, I have set up a spreadsheet where I've given a list of amino acids from 12 to 20. So if a teacher has anywhere with that number of students, they can know what list to use. But you have the students, each circle here represents an amino acid, and in this activity, each circle would also represent a student. And so the student will get an index card with an amino acid named on it. They'll draw the R side chain on one side of the card, and then they'll stand with their right hand on the shoulder of the student in front of them, and they'll hold in their left hand the amino acid index card. And then I'll have some questions for them like, well, disulfide bonds are really strong attractions between certain parts of a protein, and what kind of amino acids would cause that? And they'll, oh, I've got cysteine, it's got sulfur on it. And so I've got cysteine. It's okay, well, how can we, keeping our hands on shoulders and staying together, how can we manipulate this group and get those two cysteines together? And have them do that, staple the two index cards, and then talk about the polar connections between the positive basic amino acids and the negative acidic amino acids. And then even talking about the hydrophobic ones and how they're afraid of water and they want to be protected by those polar amino acids and how they would fold inside to try to get away from water. And so the group would manipulate themselves and kind of physically feel how this protein would be changing because of those attractions. And then that's probably only going to take part of the class period. So after they do that activity, we would have some conversation and discussion, talk about what are some of the limitations, what makes this a little less than exactly what happens in nature, but also then give them some time to start working on either their memes or their wanted posters and also their visualizations for that, like the GIFs or the videos for that formation. Then the next day is kind of the culminating activity, and that's demonstrated here, and you're welcome to try some of these protein strips if you want to. And that is going to be testing a variety of different protein-containing liquids, and they will be given dairy milk, some different plant-based milks, and some protein drinks. And then I'm going to encourage each of them to bring something from home that they know or they think contains protein and have them bring it in so they can test that. And we'll dilute them all to the same level. One of the benefits of doing that this summer is finding that when we did them at 100% concentration with these protein strips, everything showed almost maximum amount. So you do have to dilute them down to the correct level. At 50%, you started to see a little bit more color differences, and then at 25%, you started to see even a little bit more. I think looking at the numbers and doing some calculations, probably about a 10% concentration would be best. And so that's what I'm going to try with my classes in this upcoming school year. And I think I did about a 20% concentration here. So if you want to try it, we can see what it comes out. I didn't want to overfill the containers, and I didn't have a graduated cylinder to make it precise, but I tried to eyeball it to about that percent. And they'll be making the measurements with the protein strips. It has a scale on here that shows if it's this color, then it's this many grams per liter, and if this color, it's that many grams per liter. So they'll be reporting that data based on their lab experiments. And then they will look at the nutritional labels afterwards to see, well, what does the label say it has? And they'll do a little bit of dimensional analysis calculation to figure out, well, according to the label, what is it in grams per liter? And then they will make bar graphs for both, according to their wet lab data, what does it show? And then according to the nutritional labels, what do the bar graphs look like? How do they rank? Which ones have the most protein? And if you're trying to get a protein-rich diet to build those muscles, which ones should be consumed? And so that will be part of their presentation as well. The culminating assessment will be a presentation with their groups, and the groups that they make when they connect their amino acids in the bond day, that will be the same group that makes that visualization together, and also the same group that does this lab together, and the same group that does the presentation together. So on that third day, once they've made their groups, they'll stick together to do it and do it every time. And the things they'll be presenting, one is that choose one, whether it's their wanted poster or their dad joke or their pickup line, school appropriate, of course, whatever they choose. And then they'll also give their visual demonstration, the GIF or the stop motion video. They'll also show the graphs of their data, both from the wet lab portion and the nutritional calculations. And then they'll also do the L part of the KWL, what did you learn? And what questions beyond the ones we were trying to answer in the class, maybe what other questions did you answer that you wanted to figure out as we went through this unit? So that would be how we wrap it up. So that's my plan. And I'd love to thank Sam. He was actually kind enough to meet with me again this year and gave me a lot of help again this summer in curriculum. He used to be a physics teacher, so that was nice to talk to him. And also Dr. Shell and Dr. Joan Emmett for letting me participate last year in the research. For Frank and Marilyn for all the assistance and support you've given us. And Laura and Nicole, also just the feedback and the friendship that we've been able to have these last 2 summers. It's been great. So thank you very much. And do you have any questions?
Well, first of all, it's awesome. Have you ever heard of Quick Food?
Yes. I haven't used it, but I've heard of it.
But I mean, there's a learning curve to try to figure it out. But I think that would be interesting for them on as a choice on their little video.
It's flipped right out again. Okay, she can do it.
I'm not saying that you have to, but
Yeah,
I mean, the kids
it'd be
actually
interesting to
really
try.
like to use it.
Especially if they're using another classes.
Yeah.
Good idea.
I just thought that I just wanted to like 2 weeks ago.
You have that presentation that kind of summarizes your research with Sam, and will you, I believe you said you will give that this year. Are you going to do it before you get into this?
So in my class that's going to be going through this unit, it'll be the opening day of the unit.
Cool.
So, and I had a chance to practice that last year. Janelle asked me to present it to her biology classes, and then some of the other biology teachers said, well, can my students come? And then some of the other teachers said, well, can my students come? And so I ended up presenting to around 300 or 400 students. Just was a 40-minute presentation, and so I had to make it for, a lot of them were freshman biology, but they were all the way up to senior AP bio kids too. And so, yeah, I tried to make a presentation that suited all of them, and they really seemed engaged. They had a lot of feedback from the students. It was really positive. So it was good to practice that. And I know that works for the AP, so I'll be doing something similar.
You know, you were just showing us a snippet from that presentation, which was the animation of the water molecules, and there was a protein, and it was COVID, right? Or
yeah,
what,
yeah.
And
it
was a spike protein. Mm-hmm. And I was just thinking here, we've been talking all summer about proteins, but you know that was really cool because it shows some of this motion, and the protein is really vibrating, sort of, or waving around, and that is important because proteins have function, and sometimes our enzymes need to accept a substrate or something like that. I mean, do you have any just comments or thoughts on like that? You know, some of the other animations, the molecules kind of look static. I mean, is there anything that students can kind of get a sense of that motion? It's interesting.
Yeah, I only showed you one of the tutorials on Fold It, but that's actually one of the really great benefits of Fold It. As you get further into it, it starts showing you more and more complex proteins, and you start seeing alpha helices and beta pleats, and they're all in the wrong shape. And then you can move them yourself, or there's a tool called the wiggle tool, and there's a tool called the shake tool. And if you shake it, it just shakes the one strand and just kind of lets those amino acid side chains manipulate. But if you wiggle it, it wiggles the whole thing. And so you can actually see it moving as it would in water and changing shape and things coming in together and trying to eliminate those voids where water might get trapped, or some of those clashes where you would see like positives next to positives and things like that. So it actually helps them kind of visualize. They manipulate it, but if they get stuck, they can use those tools to kind of let it do it for them or do part of it for them. And so they can see it kind of in action. And it does a good job of explaining it's because they're in a watery environment. And so they're constantly moving and changing.
That's good. Yeah, that's great.
Yeah.
Yeah.
For that little chip.
Thank you.
Sorry about that. We need to take the same class.
In my last school, I was teaching only physics. And so I wouldn't have prepared this for that school. But by the time I was here, I already knew I was going to the school. I'll be teaching general chemistry, AP chemistry, general physics, AP physics. So this will be in my general chemistry classes. But I was going to tell you this, but I'll just say real quickly, if the grant for Dr. Balakrishna comes through, my principal's really excited about me doing that with AP Calc, AP Bio, AP Physics, AP Chemistry, because after the AP test, they still have a month of school. And so they always do something project-based. And so he thought, wow, we could maybe do this cooperation with her project because it's a math science integration. And so I'm really hoping that grant comes through and we can work that out because we can have lots of kids in the school participating in the same project, but all the little twists, you know, the math focus versus the science focus. So I'm hoping that works out.
If the grant does not come through, so they don't do this in
That's usually academic.
I want to trust it. Yeah. You know, previous.
Mhm. Well, we've got some things to play with, right,
Yeah.
up here?
If you want
And
to use the protein test, you want to build some models.
I've-
I wouldn't recommend eating the marshmallows, but you can.
I've got the handouts we can look at as well. Thank you.
You know, thank you.
Awesome job.
You're an incredible speaker.
Mhm.
You all are.